Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Simplifying Assay Development with Molecular Standards. Remove culturing from the equation, presented by Mr. Kyle Young. Mr. Young is a product line business specialist and a scientist at ATCC. In this presentation, Mr. Young will provide an overview of the current challenges in the development of molecular-based assays. A special focus will be placed on ATCC's wide range of molecular standards, from human pathogens to microbiome components. Mr. Young will then provide application data demonstrating the potential use of these products in diagnostic assay development. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Kyle Young.
Thank you, Brian, and thank you everyone for joining us today. This webinar is going to touch briefly on the importance of using standards for assay development and as run controls, and I'll talk a bit about how ATCC's molecular standards fill those needs. I'll go into some details about the history and design of our standards, give an overview of the standards that are available and a few that will be available soon, and I'll present some of the validation data that ATCC has generated. But first, some background on ATCC. We were founded in 1925, and we are a private, independent, nonprofit organization headquartered in Manassas, Virginia, with an R&D facility in Gaithersburg, Maryland. ATCC is the world's premier biological materials resource and standards development organization. We collaborate with and support the scientific community with industry standard products and innovative solutions. And we have an enormous range of products that include cell lines, organoids, microorganisms, various derivatives, such as GFP or luciferase tagged organisms, nucleic acids, and high titer viruses. We have microbiome standards, media and reagents, and the focus of today's talk, molecular standards. Each of our biomaterials are maintained at low passage and are authenticated and characterized using genotypic, phenotypic, and functional assays, so you have a credible starting point for your research. Let's begin with the basic importance of molecular standards. We all value having a ruler because having a standard measurement allows comparison. Molecular biology is just the same. We need a standard to compare our results or to make claims about our assay. The importance here is emphasized because of the growing use of nucleic acid amplification tests in diagnostic and surveillance assays, and in research too. Even with this increasing stature of next generation sequencing, targeted assays are still important for applications where you need to find the needle in the haystack. When designing an assay, the first use of a standard is to ensure basic functionality. This can be a pretty quick step now with the sequence data that's available, but it can be an expensive one to verify if an extensive inclusivity-exclusivity data set is necessary. Depending on the number of organisms involved, there can be as much culturing and extraction work as there is molecular work. Culture and nucleic acid extraction are expensive and time-consuming, and they can be doubly time-consuming in the case of a contamination event or a culture loss. So they can delay the actual development work, which delays manuscript submissions or product launches. The compound effect is that those extra steps not only cost money, but they delay the payoff of the work. After the assay is functional, there's lots of optimization testing to make sure that it works consistently and to try to drill the limit of detection as low as possible. And then last, the assay graduates out of the lab as it's published or goes to a beta tester in advance of a product launch. At this point, a lot of work has gone into making sure that it works reliably in-house, but the transition to another lab is a big step. I've been on both sides of a failed assay handoff where my design didn't work in another lab, or where I tried an assay that was given to me or that I found in literature and it didn't work in my hands. But probably more commonly, the assay works, but not quite the same, and some more optimization is needed. And in that case, you want to catch the differences as quickly as possible, because if they're not caught, performance discrepancies become a frustrating drain on time, money, and morale. So ATCC has two problems here that we can solve: the cost of culture and extraction, and the cost of suboptimal assays. We have the broad collection, we have the expertise to culture the organisms, we have the capability to extract them in large quantities, and we have processes in place to minimize lot-to-lot variation. But before we could get to molecular standards, we had to solve a problem of our own, and that was how to get unculturable organisms for a culture collection. In 2012, ATCC began a larger transformation to try and become a standards organization. We decided to do this because we saw the limits of what a culture collection could be, and we wanted to do more. This has guided a lot of work for us in the intervening years, including ISO certifications, a currently ongoing construction of a GMP facility, and a closer look at the materials that we offer. And in doing this last point, we identified unculturable organisms and assay controls as points where we should do more. And so in 2013, we launched the first 4 molecular standards. At that time, ATCC had only older West Nile virus strains. So as the virus began to spread in North America, we wanted to be sure we had material that was relevant for surveillance. We also chose 2 genotypes of norovirus, which is notoriously efficient at replication in vivo and notoriously difficult to culture in vitro. And Enterococcus faecalis, extracted from ATCC item number 29212, which is cited by the EPA for water quality testing. I'll come back to the noroviruses in a couple slides after we're done with the introduction. Returning to the broader view of ATCC's molecular standards, these are DNA or RNA items that are quantified by ddPCR, are backed by ATCC's extensive quality control protocols, and with a few exceptions, are downgraded to BSL-1. They save your lab the time and trouble of culturing and extracting nucleic acids, and they help prevent failed assay transfers. We have 2 basic flavors: synthetic standards, which are de novo constructs of DNA or RNA, and genomic standards, which are extracted from genuine ATCC cultures. The synthetics make possible standards for organisms that are difficult or impossible for many labs to culture, either because the organism doesn't grow in vitro or because it requires a high containment level. On the other side, the genomic standards are extracted directly from ATCC's source material and contain the full organism genome. An additional benefit coming soon for the genomic standards is that many of them will be included in our enhanced authentication initiative. Our portal for the EAI is going live on Monday, September 30th at genomes.atcc.org, where full sequence data for ATCC cultures will be available and searchable for free. We're starting with 250 bacterial genomes, but we'll be adding more every month, and all the genomic standards will eventually be sequenced. And just quickly before I move on, some of the basic applications for these standards are to generate a standard curve for qPCR to determine bacterial, viral, fungal, or protist load, as a control for molecular diagnostic assay development, verification, and validation, to monitor assay to assay and lot to lot variations, and to assign a genome copy number to secondary calibrators, for example, to establish a ratio of plaque forming or colony forming units to genome copies. The basic specifications of the 2 types of standards are similar. The synthetics are fully sequenced to verify the construct matches our design, and we test them using at least 1 published qPCR assay, which we report on our website. The genomic standards are authenticated by amplicon sequencing, and the gene target varies for different organisms, and the nucleic acid integrity is verified by gel electrophoresis. Each standard type is filled with 100 microliters at 10 to the 5 to 10 to the 6 genome copies per microliter, with a specific genome count reported on the certificate of analysis, and both types are shipped frozen. Now I'll go into a little bit more depth into the design process of the synthetic standards. And I'll start with some improvements that we made to the first generation. While the initial interest in the standards was high, we found that there were some facets that could be better. Armed with lots of constructive feedback, we redesigned the norovirus constructs and presented the changes in a poster at the Clinical Virology Symposium in 2015. First, We changed the preservation format from dried to frozen and added an RNA stabilizer. These first two changes increased the shelf life dramatically. We added droplet digital PCR quantitation for a more accurate genome copy count. We began manufacturing with ISO 13485 compliance, and we expanded the construct by adding a portion of the RNA-dependent RNA polymerase gene to increase the number of published primer sets that would bind. Before I move on, ATCC is still trying to improve, and I'd like to invite your feedback on any of the molecular standards that you've worked with or would like to see us make. Our sequencing project is a big step, but we're looking for other steps to take too. If you have an idea that you'd like us to know about, please send me an email at kyoung@atcc.org. So with two sets of constructs, the obvious next step is to compare them, and that's what we did. The next several slides are from the poster that we presented at CVS. We chose two primer and probe sets, one from CaliciNet, that's CDC's norovirus outbreak surveillance network that includes public health laboratories from every level of the US government, and another recommended by the European Committee for Standards, the ECS working group. This slide shows the standard curves used using the CaliciNet primers and probes for the new norovirus G1 in the upper panels and the norovirus G2 construct in the lower panels. For both constructs, we have a nice linear progression between dilutions. We ran the ECS assay as well with similarly good results, which are reported in the breakout box at right. Next, a direct comparison of the old and new constructs using the CaliciNet and ECS working group primer and probe sets, with the new constructs in blue and the old in green. For norovirus genotype 1, you can see on the upper panels that the new construct performs slightly better than the old one using the CaliciNet assay. And on the lower panels that it outperforms the old one significantly when using the ECS working groups assay, where the primers bound poorly or not at all in the first construct. Genotype 2 was also tested in this way, again shown with the CaliciNet assay in the upper panels and the ECS in the lower panels. And the new construct shows a slight leftward shift in the amplification curve for both, indicating that it's a bit more efficient than the old construct for both assays. Last, we used the respective standard curves to quantify the working reagents for norovirus genotypes 1 and 2 from the National Institute for Biological Standards and Control, NIBSC. Here, sample amplification plots are shown with the standard curves in blue and NIBSC working reagent dilutions are in red. The working reagents were assayed in triplicate using each assay at undiluted 1 to 10 and 1 to 100 dilutions. Our results show that there were 2.3 times 10 to the third genome copies per microliter in the norovirus 1 reagent and 2.4 times 10 to the 2 genome copies per microliter in the norovirus 2 reagent. So gathering all that we learned from our initial design and then the redesign, we constructed the following basic process for making synthetic standards. First, we vet the need for the construct based on public health concern, volume of published research, and direct feedback from the research community. Then we collect as much relevant data as we can from public sources in order to assemble a draft construct. Our designer creates and analyzes a number of phylogenetic trees and sequence alignments to determine the most valuable binding sites to include. After that, we have one last technical review to finalize the construct. Following completion of the design, we have a regulatory review to make sure we don't make anything that could be used nefariously. And then the construct is actually produced. Assays are double checked and optimized, and last, vials are filled and tested. There are a number of questions that we commonly field at conferences and through our technical service department. And before I move on, I'd like to answer a few of them. The synthetic standards are small, typically less than 5,000 nucleotides in length. Because of their size, in most cases, they do not include entire genes, only gene fragments. There's enough sequence there for assays to function, but the amplicon size may not always match what you'd find from a wild-type genome. Our choice of nucleic acids is made to match the natural genome, so there are several RNA constructs like the norovirus standards that we already discussed. We recognize that you need a starting point for your work, so we do report an assay that we've confirmed to work on the website. Other assays can be determined by looking at the gene fragments included in the construct, or at the references on the website. The actual sequence, though, is proprietary, so we can't disclose it, but we're happy to confirm whether your primers and probe will bind to it. With that, let's take a quick look through the standards that we offer. There are a lot of organisms on this slide, so I'm going to pause on it for a few minutes to give an overview. The link at top right will take you to a landing page on the ATCC website with more information about the standards. From the 4 initial standards, we've grown to 159 total items, 115 genomic, and 44 synthetic. As you saw earlier, the first 4 molecular standards were all human pathogens, and we've done a lot of work making more standards for pathogens of various types. We have bloodborne pathogens such as HIV-1 and hepatitis C, gastrointestinal pathogens like astrovirus, cryptosporidium parvum, salmonella, and E. coli, respiratory pathogens including coronaviruses and several species of mycobacterium, STIs such as human papillomavirus, herpes viruses, and chlamydia, vector-borne pathogens like dengue virus and West Nile virus, and epidermal and nosocomial pathogens like Staphylococcus aureus and Candida albicans. We're also beginning to add standards for organisms found in the human microbiome, broken out here by location. There is some overlap with the pathogen slide, but many of these do not necessarily cause disease in healthy adults. This is the area where we are most actively adding standards right now. One of the first standards we released was 29212Q-FZ, which is for water safety testing. You'll notice the suffix is different. This item was made using an older developmental process, and it's back in the pipeline for production as a DQ product. We've also added a number of other standards for organisms that are cited by the EPA or other bodies for water safety testing. Last, as far as our current groups for standards is food safety standards. Norovirus G1 and G2 are the 2 most common genogroups of norovirus to cause human infection. We have a murine norovirus synthetic standard to support laboratory research on norovirus. This was made as a synthetic because, like many organizations, ATCC has difficulty making pathogens in vivo. We also have the Big 6 E. coli strains, which are available as a panel in item number MP-10. Most recently, we launched 5 standards for pathogens in August, including a synthetic human herpes virus 7 and genomic Trichomonas vaginalis, Chlamydophila pneumoniae, Pseudomonas aeruginosa, and Bordetella peripertussis. And looking forward, there's about 20 to 25 more standards that we hope to launch in 2019. But for the sake of accurate reporting, I've only listed those that we expect to launch in the next several weeks. So if all goes well today, ATCC will launch the top 6 items from this list tomorrow. In addition to the species listed here, one notable new branch in our direction is a group of Aspergillus species for testing agricultural products. That covers the high points of the collection, so let's look at some more validation data. And I'll start with some research that ATCC did in support of a poster we brought to the Clinical Virology Symposium earlier this year. Just a quick note first. Hepatitis viruses are named for the disease that they cause, based on infection of the liver. Other than that, they have very little in common, including their classification, which places them in different families of viruses. For example, hepatitis B is a member of Hepadnaviridae orthohepadnavirus and has a DNA genome, while hepatitis C is a member of Flaviviridae hepacivirus and has an RNA genome. Some other hepatitis viruses include hepatitis A, which is a Picornaviridae hepatovirus, for which we also have viral stocks, hepatitis D, and hepatitis E. This project was similar in many ways to the norovirus redesign follow-up analysis. So first, we generated a standard curve using our hepatitis B and C virus standards. On this slide, you see the hepatitis B qPCR curve, which ranges from 3 × 10⁵ copies to 3 copies in serial 1 to 10 dilutions. The NIBSC hepatitis B working reagent, code 10/264, was reconstituted in 0.5 milliliters of molecular grade water and extracted using a commercially available kit. Only one curve is shown, but this assay was performed 3 times with each set run in triplicate, with each sample run in triplicate, with excellent linearity by R-squared, slope, and CT spacing. We did the same for the hepatitis C standard. The standard curve for qRT-PCR was set from 1 × 10⁵ copies to 10 copies by serial 1:10 dilution. The NIBSC hepatitis C working reagent, code 06/102, was reconstituted in 0.5 milliliters of molecular grade water and extracted in the same way as HBV. Each dilution was run in triplicate, and the assay was repeated 3 times, again with very good linearity. For this project, we also ran ddPCR. This slide shows the one-dimensional amplitude scatter plots of positive and negative digital droplet PCR for 3 dilutions from 10⁴ to 10⁶ copies in triplicate for HPV on the left and HCV on the right. This assay used the same primers as the qPCR and qRT-PCR. Here we're also showing a consistent progression between dilutions. Because the NIBSC reports infectious units per milliliter, we were able to calculate the number of genome copies in an infectious unit as being 11.4 genome copies per IU for the HBV and 61.5 genome copies per IU for the HCV reagent. Last, I want to share some data that we generated for our human herpes virus 1 and 2 genomic standards. The bulk of the work is the same as what I've previously shown, so I've summarized that part in one slide. The blue standard curves for HSV1 in the upper panels and HSV2 on the lower panels range from 2 × 10⁵ copies to 2 copies by serial 1:10 dilution. Viral DNA from NIBSC, HSV1, and HSV2 working reagents was extracted using the same kit as before and run undiluted at 1:10 and at 1:100. And these are shown in red on the sample amplification curves. These qPCRs were then used to quantify the genome copies in the sample within a very narrow interval near 10⁵ genome copies per microliter. As an added element to our own HSV work, we sent vials of a non-quantitated version of the material that is prepared using many of the same processes to the Cincinnati Children's Hospital Medical Center. There, the DNA was diluted 1 to 1,000 and used in a laboratory-developed test by the pathology department over a 3-month period by 3 different operators in 36 total assays for each virus. These data were tracked and are graphed by the threshold cycle count for each assay run. We have one outlier, but overall the consistency is very good. During the test period, the HSV1 positive control had a standard deviation of 1.14 and a coefficient of variation of 3.9%. The HSV2 positive control had a standard deviation of just 0.54 and a CV of only 2%. The data I've shown today was pulled from posters that ATCC presented at the Clinical Virology Symposium in 2015, 2016, and 2019. We've also presented posters for our dengue virus standards in 2014, our Zika virus standard in 2017, and our human parechovirus 3 standard in 2018. All of these can be found on our website using the URL at the bottom of the slide or by hovering over resources in the ATCC website ribbon and then clicking on posters and presentations under the Learning Center section. And if you missed us at CVS or ASM this year, but you want a chance to meet us in person for more information, ATCC will be sending representatives to AMP and ASTMH in November. So we hope to meet you at our booth. In summary, ATCC molecular standards are a consistent, reliable starting material for assay development and control that eliminate the cost of growth and extraction. ATCC has 159 total standards, including 44 synthetic items that cover many common unculturable organisms with human health implications. And our total offering will grow by another 20 to 25 standards by the end of 2019. So last, I'd like to thank everyone who worked on this webinar, either by generating the data, creating the standards, or producing this event. So thank you to the ATCC R&D and technical transfer teams and the marketing team. Thank you also very warmly to Donna Diorio of the Cincinnati Children's Hospital, the National Institute of Biological Standards and Control in the UK, and to Dr. Pinsky at Stanford, who beta tested many of our early synthetic standards. So thank you for listening. Are there any questions?
Well, thank you, Kyle. In just a few moments, we'll begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Okay, our first question seems to be around synthetic constructs. Why are there some synthetic constructs for organisms that can be grown in a BSL-2 laboratory?
Yeah, so we've got two main reasons for that. One is just convenience. So we've got the hepatitis A. We wanted to provide a full suite, so we created that just so there would be a consistent panel of products that were suitable for investigation in that area. The other exception for that rule is if ATCC doesn't have an organism in its collection that's clinically relevant or contemporary, for example, the Zika virus standard.
Okay, and here's the next question. It's around reference genes. What reference genes do you use for bacteria, viruses, and fungi with your qPCR?
So for the qPCR for synthetics, we just used a published assay for one of the genes that we've chosen for the construct. For general quantitation, it varies organism to organism. So we try to choose, or we do choose, a single copy gene for protozoans and for bacteria. And then for viruses, we try to choose something that's going to be conserved as the virus's passage.
Okay, I guess obviously we're going to be doing this for a while, making these standards. What standards is ATCC going to make in the future?
So we're continuing to extend the suite of pathogens that we have, and specifically we're going to work on water safety testing and food safety testing and agricultural safety standards. We're also looking at microbiome and probiotic standards. And once again, just to reiterate as I said in the webinar, if there's a standard that you'd like us to make, please send us an email, or you can contact me directly at kyyoung@atcc.org.
Okay. We've got another question in that's around biosafety levels. You mentioned some exceptions to the BSL-1 downgrade. What are they, and then why were they done?
So there were about 10 or 11 respiratory viruses that were not downgraded and are still at BSL-2. So we did this at the time with the idea that these were going to be used primarily in clinical labs that were already operating at BSL-2 and that it wouldn't be a problem. But we got really strong, fast feedback that we do need the downgrade. So there's 11 of them. All 11 are going back through for safety testing and downgrade in the near future.
Okay. Here's an interesting question around ATCC's Enhanced Authentication Initiative. So are there any of the molecular standards being included in the initiative? And also, can you maybe talk a little bit about what the Enhanced Authentication Initiative is?
Sure. So to answer the first part of that question, there's 25 genomic standards that are being included in the EAI as it's going to be launched next week. Eventually, all of the genomic standards will be included. But basically, the project is to sequence all of ATCC's catalog of items. So we're going to start with the most frequent distributors, the items that we send out the most, and work our way down. And hopefully, eventually, one day, everything will be in the online portal, and we'll be able to search the sequence of every item that ATCC has.
That's really exciting, actually. We're all pretty excited for this to happen. Now, how are the genomic standards extracted?
So the specific extraction method is something that we need to keep confidential, but in general, we use classical methods for fungi, bacteria, and protists, and we use a commercially available kit for viruses.
Okay. Now, here's a question that just came in. Can you revisit the HCM IU per mil numbers? And how was that copy conversion made?
So basically, the WHO standards come with IU per mil reported, and then we calculated the genome copies per mil and just uses the volume as the common denominator to figure out the number of genome copies per IU.
Okay. Why was this item 29212Q-FZ, why is it being reworked?
So basically that item turned out overcomplicated and very expensive. So it came with several dilutions prepackaged that each needed QC testing, and it was made at a time when our cost for accurate molecular quantitation was much higher. So we are simplifying it now to align with the other genomic standards, and we're going to relaunch it as a member of the DQ product line at a lower price.
So basically, we're just making a better product then, right? Okay, so can ATCC make a custom standard?
Yes, we can make custom standards. You can order one by contacting us at ATCC bioservices@atcc.org.
Okay, here's another question that's just come in. How do you guarantee the genome did not change after a few generations? So I guess genetic drift.
Is this in reference to the Enhanced Authentication Initiative? I think that must be what they're asking about.
It's okay to text in some clarification around that. But.
So if this is about the sequencing project that we have going, I don't think we're making that guarantee now. We would like to do this as a lot specific quality control test, but we don't have the capability as of now. So we are sequencing items where they are at the current passage, and we can't make any guarantees about genomic sequence of a live organism.
Okay, but you know, we do use the seed stock principle, right? Which generally means that the organism that's distributed is going to be as close to the original organism as possible.
Right. So we have some assurances, but no guarantees.
Okay. All right. Now that's all reasonable. That's reasonable. Now here's a question that I have. Okay. What's a DQ product and what's an SD product?
Okay, right. So we did skip over this, didn't we? So the suffix to the item number tells you, well, what kind of derivative it is. So SD stands for synthetic DNA. A D or a D5 it just stands for DNA. The D5 is 5 micrograms. A DQ is quantitative DNA. And we carry that over to RNA products as well, just for uniformity.
Okay. All right. Well, at this time, we will conclude our Q&A session. I'd like to thank Kyle for the excellent presentation, and thank you everyone for attending the webinar. Please join us for more webinars in the ATCC Excellence in Research webinar series. So on October 24th, Diana Douglas will present ATCC's new epithelial mesenchymal transition reporter models, which elevate biological models of metastasis. And then on October 31st, Dr. Siddhartha Paul will present his talk on exosomes on the edge of the bubble, use of exosomes as reference materials in biomedical research. Thank you again, and have a great day.